This vodcast is an overview of cellular respiration. Spent the last few vodcasts going through the steps of cellular respiration, which were glycolysis, the citric acid cycle, and then finally the electron transport chain and oxidative phosphorylation. So now we're going to put it all together into one kind of view or one idea, and make sure that you can see this as a whole and not its individual steps to see our outcome of how much ATP is made when all of cellular respiration is done. So you should have a little handout that looks somewhat like this. It's not an exact match, but I think you should be able to follow along with it as I go through this overview. We're going to go through each of the steps one at a time and make sure that you can see what the main products are and how they result in the formation of our ATP. On your diagram, you might want to label a few things to begin with. Outside of this yellow bean structure is the cytoplasm, so that's what this box here is for. So that's the cytoplasm of the cell. And this box over here is labeling the actual structure where cellular respiration in the cell takes place, that being the mitochondria. Once we've got those locations labeled, we're going to label the main processes. So glycolysis happens outside of the mitochondria in the cytoplasm. The Krebs cycle happens, remember, in the matrix of the mitochondria, so within all these folds, the very inside fluid of the mitochondria. And then finally, the electron transport chain and oxidative phosphorylation occurs across this inner membrane. And on both sides of that, one side being the matrix and the other side being the inner membrane space. Alright, so that's where the electron transport chain takes place. Okay, so now that we've got all of our locations and different steps taken care of, we're going to start figuring out what they make. So we're taking a look at glycolysis first. You have your diagram for glycolysis already figured out. This is what it should look like. We've written that down earlier. So again, you start with the glucose here. You end up with 2-pyruvate at the end. But in the process, you do make some products. So let's look at our ATP first. Okay, ATP is used here as well as here. So when we turn glucose into G3P, we're actually at a -2 on our ATP count. But then when you turn G3P into pyruvate, each of those makes 2 ATP, so that's a +4 on our ATP count. So the total for our ATP is 2 ATP made in glycolysis. The other products we make in glycolysis are the NADH. One NADH, or hydrogen ion carrier, is made here, and the other made here, for a total of 2 NADHs made in glycolysis. So we're going to add those to the diagram we were just on. Here's glycolysis. Remember, in the end, 2 pyruvate are made, so fill that in first. So 2 for your pyruvate. How many NADHs were made from glycolysis? That's what this was here. Remember, we just calculated that was 2. And then how many ATPs are made in glycolysis? That's what is here. Again, we just calculated that was 2. So far we've made 2 NADHs and 2 net ATPs. Okay, next we're going to go on to the citric acid cycle, which is sometimes called the Krebs cycle, and look at what we get from products from that reaction. Okay, this is what your diagram should look like for this process. We're going to start with this little conversion, up at the top, where we change the pyruvate into acetyl-CoA. right there, because we do have some products from that reaction. Now remember, we're going to do this per glucose, so the citric acid cycle is actually happening twice. So all these numbers we calculate are times 2. So first we've got some NADH made. That's right here. There's 1 made, but since it's times 2, we've got 2 NADHs made per glucose. Remember, every glucose ends up being 2 pyruvates, so these 2 pyruvates are going to end up making 2 NADHs. So we're going to add that to our diagram. Clicked on the wrong spot there. Alright, so as we change that pyruvate, there are 2 of them made, into acetyl-CoA, and again there are 2 of these made, 1 per pyruvate, we make some NADH. Follow the line up, 2 NADH. Okay, now we can get back into this citric acid cycle. We're going to again look for ATPs and NADHs. So remember, this turns twice, so we're going to take everything times 2. NADH made here, NADH made here, NADH made here. That's 3. 3 times 2 is 6. So 6 NADHs. Next step we're going to look for is ATPs. We have 1 ATP made here times 2 equals 2. And the last product we're interested in the citric acid cycle are these FADH2s. And again, we have 1 made times 2 equals 2 total. So we've got 6 NADHs, 2 ATPs, and 2 FADH2s made in the citric acid cycle per glucose. That's why we multiplied everything by 10, or by 2. Okay, let's go back to our main diagram and add those in. So again, here's our citric acid cycle. Products that come out of it are up here, up above. So we want to label how many NADHs we made. Remember what we just talked about? The NADH number was 6. We also want to label how many FADH2s we made. That number was 2. And then the ATPs that come out of the citric acid cycle are down here. And remember, we made 2 ATPs. So far, the 2 ATPs in glycolysis plus the 2 ATPs from the citric acid cycle give us 4 total for the reaction. And now we're going to get into the major function of producing ATPs for the mitochondria, which is in oxidative phosphorylation. So we made all of these NADHs. Remember, there were 2 here and 2 here and 6 here and 2 here. They dropped out their hydrogen ions, flowed through the membrane, flowed back, helped to make the enzyme get activated that makes the ATP. So we're going to use all of those hydrogen ion carriers to make ATP down here. And let's see how much we end up with. Again, I just want to review with you to make sure you remember this process. I'm going to jump to what you should have for oxidative phosphorylation. Here are the NADHs and the FADH2s I was just talking about. Remember, we had 2 plus 2 plus 6 NADHs, so there's actually 10. And the FADH2, there were 2 of those. They actually drop off their hydrogens here in the matrix. The hydrogens get pumped across through active transport on those electron carriers. They build up in concentration in the intermembrane space, flow through protein channels, activating ATP synthase to make 32 ATPs. In the meantime, the electrons from those molecules from the NADH and the FADH2 are getting transported between these carriers to help pump the hydrogen ions through the channels. The final electron carrier is oxygen. It combines with water, or combines with hydrogen ions to produce water as a byproduct. Okay, kind of like a waste product of of the electron transport chain. So we're going to add this into our total equation on our summary, and 32 ATP is the main thing because there are no NADHs or FADH2s made here. So right down here, did it again. Sorry. So right down here, and how many ATPs are made in the electron transport chain? We can fill in 32. So 32 there, 2 from citric acid, 2 from glycolysis, gives our final number of about 36 ATP per glucose molecule. Alright, so that's the whole process all together there. Remember, we built up a bunch of these hydrogen ion carriers so that they could activate the enzyme in the electron transport chain that made the majority of the ATPs. A few are made in the Krebs cycle and in glycolysis, but the main one that's very productive is oxidative phosphorylation.